Hey guys, so we're starting chapter 2. We're going to look more at what it looks like when things are microscopic. We've been talking about the things that we can see, like beetles and snakes and wolves, and looking at point Peely, but what about the things that are harder to see or not visible at all to our eye? So we're going to talk about how the simple can actually become complex. This is a rock formation. It's found in a lake in British Columbia. They call it a microbiolate, and it's covered with different types of bacteria and other microorganisms that actually trap minerals from the water to form the solid stone structure that it lives on. So that stone structure is made up of bacteria and microorganisms. They used to exist millions of years ago a lot, and now they don't so much. So scientists will actually use that to look at the inside of the history of the earth and study different life forms, even at the microscopic level. Just food for thought, like what does this kind of thing suggest about evolutionary history of bacterial cells, if this has been around for millions of years and these cells have figured out how to do this? Sort of, do they communicate, or have they evolved? Just some food for thought. Just a recall on prokaryotic cells versus eukaryotic cells. They differ a lot on their size and structure and a lot of other characteristics. Bacteria and archaea are prokaryotes. They are not membrane bound. They're very simple structured, very small. Protists, plants, fungi, and animals are eukaryotes. They're membrane bound, complex, and 1,000 times larger than their prokaryotic neighbors. Viruses are a whole different ballgame, and forgive me because I think they're really cool, and I can understand why we get stumped as scientists trying to figure out solutions to them. They're functionally dependent on a prokaryotic or eukaryotic cell. Viruses cannot function alone. They cannot live outside of a cell alone. They have to invade another cell. And what they do is they invade, and then they use the host cell's machinery, so how it replicates its DNA, to survive as well as to reproduce. They are not cellular. They are not prokaryotic. They are not eukaryotic. They have no cytoplasm, no membrane-bound organelles, no cell membranes. And they cause disease in the host cell. Different types of plant infections, HIV is a virus, polio, SARS-CoV-2, which is actually COVID-19, is a virus. When we classify viruses, we do so by their unique characteristics. They all differ in size and shape of the capsid. So that figure right there that you're looking at, A, B, C, and D, those are different shapes of the virus and their structures. So the first one, A, is the poliovirus. B is what HIV looks like. C is a tobacco mosaic virus. And D is the T4 virus. So you can see how they're all very different in sort of their morphology, if that's what we're talking about, and the way that they look and their shape and their structure. They also are different types of diseases that the virus cause, like polio and HIV. Inside their genome, or their set of genes, is the genetic material of RNA and DNA, so that can be sequenced for analysis and classification as well, and the way that they reproduce. So you can see this is sort of similar to what we've talked about with the other things, the structure, their genetic material, how they reproduce. So how does a virus actually reproduce if it's not a cell? Well, this is where it becomes dependent on its host cell. So they don't, in fact, reproduce or divide. What they do is they replicate. Some viruses use a replication process called the lytic cycle. So what it does is it quickly attaches to a cell, it enters that cell, it replicates its DNA, it assembles, I can see that assembles is actually spelled wrong, and releases from the cell, killing that cell in the process. When the virus is released from that cell, it now can go infect other neighboring cells. Other viruses replicate by using the lysogenic cycle. I think of this as sort of going dormant at times and not being active. So it'll enter and attach their DNA to the host's chromosome in the nucleus, meaning that it is now a part of that cell. It is not going to be released, but for every cell that cell now divides into, will always have a piece of that virus DNA. It's referred to as a provirus and can remain dormant in a host and reactivate later as a lytic cycle. So when it's a virus that can go lysogenic, it can also use the lytic cycle. And proviruses do not kill host cells. So we don't damage any of the good cells. They just sort of go along for the ride and stay inside of the DNA of the host. And when I say host, it could be anything. It could be a human, a cow, a plant, any sort of cell. This picture is actually in your textbook. It's on page 55. I highly recommend you go look at it. It has a breakdown. I know some of you will copy it. If some of you can figure out a way to copy it, it's a good idea. It just looks at how the two different cycles work. Please note that this is going counterclockwise, so pay attention to the arrows. So it starts at attachment where it actually meets the cell, the blue being the cell, the sort of yellow stick is the virus. And then it will enter by injecting its genetic material into that host cell. Moving through the lytic cell, the host cell will then replicate everything that the virus needs. Inside the cell, the virus will assemble, and it will cause the cell to lyse or burst and release all that new virus. So instead of now just one attaching to one cell, I've made 3, and now I can go infect 3 other cells, and so on and so on. Looking at it from the lytic cycle, when it enters into the cell, it actually goes in and causes the cell's DNA to pick up that DNA and constantly make it. So it's constantly in all the cells. You can see that this cell does not lyse in the lysogenic cycle. It can then transition into the lytic cycle where it does cause infection and death. We'll talk about a couple viruses that do both. Virus and disease. So lytic viruses, they burst the cell and infect the neighboring cells, and already damaged host organisms will be more rapidly affected. So we talk about immunocompromised. This is very common now as we talk about those who are sort of susceptible to COVID-19 because they may be elderly, so they're already immunocompromised. They may be cancer patients or cancer survivors. They may have underlying causes like heart conditions or conditions with their lungs like COPD. And those people are already working with cells that are weaker. So therefore, they get affected more rapidly because they're sort of outnumbered. Lysogenic viruses, they may not have an immediate effect on a host organism. So example is HIV is a lysogenic virus and it's a retrovirus. So what it does is, in this picture, you can see all the little blue spots. Those are HIV viruses attaching to this cell to cause it to pick up its genetic information and continue to pass it on to all the other cells in the body. What's a retrovirus? So a retrovirus actually carries RNA and an enzyme inside it called reverse transcriptase. That causes the host cell to copy this information all the time. It then embeds it into the host chromosome and becomes a provirus so that every descendant cell has HIV or DNA copies within its genome. Looking at patterns of diseases, as we can see these two cell types. So you're looking at the bright orange is herpes simplex virus, and then the more purple blue is the human immunodeficiency virus or HIV. They clearly look very different. Their receptors or those things that are sticking out of them are very different, and therefore what they do and what cells they attach to are very different. Herpes is your common cold. When it comes out of its lysogenic stage and it starts to infect, you get that cold sore. That's how you know your herpes is active. It may stay dormant in you for a very long time, and then when you get a cold sore, that's when it's sort of become active. Human immunodeficiency virus, or HIV, is in the lysogenic cycle for quite some time. People can be HIV positive and have no signs or symptoms for that reason until it moves into the lytic cycle where it starts to infect all the cells around it. And that's when you get complications and immunocompromisation. Prions are neither bacteria nor eukarya, no, not prokaryotic. They're not viruses, they're not retroviruses. They've kind of had scientists stumped for a while. So it's been observed that there are several types of brain diseases specifically that will continue to cause infection even after radiation. And the interesting thing about radiation is it destroys the DNA or the RNA. So if it were a virus who's dependent on their DNA to be replicated, then these specific diseases should be cleared with radiation because you've just cleared all the DNA. But that's not happening for some of these diseases. So what they've discovered is these diseases are actually caused by prions. And what they are is they're a protein, and they lack DNA and RNA. As a side note, what they do is they just sort of mutate and dysfunction. So mad cow disease is a prion, and then they pass on that mutated or dysfunctional protein by ingestion. So simply eating a cow that has mad cow could make you susceptible to mad cow disease. Virus is important and kind of neat for biotechnology. So if a scientist can figure out how to just harness the capabilities of the virus, it can then insert things that might be missing from a human genome and cause that to be infiltrated into the body. So instead of constantly putting in this thing that will infect and break down all the cells around you, it may be actually beneficial to make a gene that might be missing through your transcription process. And we'll talk more about genes when we get into the body systems and how things replicate and stuff like that. But for this, we're talking more about classifying the DNA. This was just an important hit point. Any questions, you feel free to ask me when we get in class, and I'll see you tomorrow.